All righty. So when we were so rudely interrupted by the interweb, we were talking about the spaces, right? Again, these are fluid-filled spaces containing cerebral spinal fluid. As we talked about, you can start in the lateral ventricles, travel from the lateral ventricle through the interventricular foramen into the third ventricle. From the third ventricle, there's only one way out, and that is down the cerebral aqueduct into the fourth ventricle. But as I was saying, from the fourth ventricle, we have choices. We have options on where we can go. Notice to the side, there is not one, but after all, since we have two sides, two lateral apertures. These two lateral apertures are located basically just above the pons. And as the name would indicate, to the sides. And as the cerebral spinal fluid exits those lateral apertures, they would enter into the subarachnoid space. But notice, and here the picture is a little bit tricky, but if you look here, there is a third aperture. This one is located on the midline, and this one is basically below the pons, and this is the median aperture. And it also enters into the subarachnoid space. But there is a fourth exit to the There is a fourth exit to the fourth ventricle as well, and that is to travel. Oops, I didn't want that to be green. I wanted that to be pink. Travel through the center of the spinal cord. As we'll see, there is a fluid-filled space at the center of the spinal cord. And that fluid-filled space down the center of the spinal cord is known as the central canal. And once it traveled all the way down to the bottom of the central canal, it would then exit into the subarachnoid space. So notice one last thing here. Even though there's four ways out of the fourth ventricle, that's convenient, four ways out, fourth ventricle, no matter which of those four routes the cerebral spinal fluid takes, it's always going to enter into the subarachnoid space. So remember, we talked about there was that big, huge fluid-filled subarachnoid space, but we said the cerebral spinal fluid wasn't made there. Instead of being made there, it is actually made here inside of the fourth ventricles in that choroid plexus. And again, remember the choroid plexus, I'll draw it one last time, in the inferior part of the lateral ventricles, here in the superior part of the third ventricle, and two parallel rows of it lining the center of the fourth ventricle. I've drawn it on the illustrations, but notice we have these other pictures that do a nice job of showing it. Notice here we have the choroid plexus in the small part of the lateral ventricle we can see. Notice from this view, we can see that interventricular foramen to go from the lateral ventricle into the third ventricle. Notice here is that choroid plexus on the superior part of the third ventricle, where it then goes through the cerebral aqueduct to the fourth ventricle between the pons and the cerebellum, where there is yet more choroid plexus. Notice here, we see that median aperture located below the pons. Notice our arrow points right here, but it'd be poking out to the sides would be our lateral apertures, basically near the top of the pons. And here we see that central canal that goes all the way down to the bottom and then out into the subarachnoid space. So here we see a couple nice illustrations showing this. I think I have, I'll talk about that one in a minute. So we have,
Professor?
Yes.
Oh, question. So if someone has hydroencephalitis and they go to do a surgery to create a shunt, where do they place that shunt and where do they shunt it to?
So part of it depends on what the problem is. If it is an overproduction, then they have a couple of different options of where they can put it. Notice the majority of the cerebral spinal fluid kind of comes out near the base of the neck. So that's not an uncommon place for them to put it, because coming out of the lateral ventricles and the median ventricle, I mean the lateral aperture and the median aperture, there's a lot of fluid that pours out into these spaces. Or sometimes they'll do it further down the spinal cord as well, because these things are all connected. So I think it more has to do with what's causing the problem as opposed to having one primary location where they would send it. But that's not an uncommon place near the base of the skull. Yes, Arthur, I think you were first.
Yeah. So we just need to know like the location. We don't really need to know the function. It's kind of straightforward.
Well, so again, we need to know the function of the cerebral spinal fluid. As we talked about, it cushions the brain and provides protection, but it also provides chemical signals, osmolarity signals, hormones, things like that, and removes toxins from the nervous tissue. So it's important that way. And then the rest of these are really just spaces or the structures that make the fluid. Yes.
Okay. Yeah.
Yulia?
Yeah, I think I missed something. So does it have to, does it have to be moving the cerebral spinal fluid in order to deliver things like nutrients, or is there another reason?
Well, it also provides that cushion as well. But you're right. If the goal was just to cushion it, then that would be something that we could just fill this space with jelly, with adipose or packing peanuts or something like that, and it would provide the cushion. So the fact that it's constantly circulating tells us that it must be serving some other function. It's not just about cushioning. And remember, this is basically filtered blood plasma. So this blood plasma that's filtered out is going to have like chemical signals like hormones, electrolytes. It's going to have the pH. It's going to have the osmolarity. So it's going to tell the brain a lot about what's going on in the blood, which obviously tells the brain a lot about what's going on in the body. In fact, we talked about this with muscles. Remember, we talked about muscle fatigue, that systemic fatigue that we have, when that lactic acid causes us to stop moving because we don't want to hurt ourselves. Well, this is where that happens. Lactic acid is released into the cerebral spinal fluid. Our nervous tissue sees that lactic acid and says, oh, we got to stop doing that muscle activity. We're going to hurt ourselves, so let's stop running. So yeah, so there's all sorts of signals and stuff that we receive from this. Yes, you had a second question?
Yeah, but that will be jumping back. I'm sorry, I think I lost it. It had something to do with hormones. Yeah, I'm sorry. It will come to me and then I'll type it.
Okay, excellent. So yes, so there's all this stuff. At any one time, we have within these spaces about 150 milliliters of cerebral spinal fluid. And during the course of a day, you make about half a liter. So we're constantly making it, constantly recycling it. However, notice we've talked about where it's made and where it flows. What we haven't talked about is how we get rid of it. And that goes back to those dural sinuses we were talking about. Notice here's that picture we saw before with the two layers of the dura, the subdural space, the arachnoid matter, the arachnoid fibers, the pia matter, and this subarachnoid space. Well, notice there is this extension of the arachnoid matter, which pushes up like a finger-like projection into the dural sinus. And this finger-like projection is called the arachnoid villi or the arachnoid trabecula. And so as we produce that cerebral spinal fluid, the cerebral spinal fluid comes into the subarachnoid space and then drains back into the veins. Remember, this dural sinus is a big blood vessel, so it came out of the blood vessel and it goes back into the blood vessel. So this is how it circulates and works its way back. Yes, go ahead.
Yeah, it was about the blood-brain barrier. Does, like when we drink alcohol, does it get into our cerebrospinal fluid or brain
No.
at all, or no?
Great question. So again, our blood-brain barrier is the capillaries between the blood vessels and the nervous tissue itself. And remember, there are astrocytes wrap around the capillaries to limit what gets out into our nervous tissue. And remember, it does that by wrapping its cell body around that capillary. Alcohol is lipid soluble. So it passes right through that blood-brain barrier and obviously affects our cognitive function. Same thing here as well. Alcohol can get into our cerebrospinal fluid as well, because here the ependymal cells are filtering what comes out. But remember, both the blood-brain barrier and these ependymal cells filter, but they don't limit everything. It's not that they stop everything from getting out. They just want to limit what gets out. We don't want big proteins. We don't want large things. We don't want, viruses or things like that to get through. So we try to limit what gets out, but plenty of important things do absolutely get out, and things that are small, things that are lipid soluble. There are plenty of things that get out that can affect nervous tissue. Yes, Arthur.
How can alcohol go through? How is lipid soluble and other ions cannot really go through?
Right. Sodium is not lipid soluble, so it has a harder time getting across a plasma membrane, right? It needs channels and things like that to get out. Alcohol is lipid soluble. So really, no matter how many layers of fat you put on the outside of a capillary, alcohol just floats right on through because it's lipid soluble. So it passes through without any problem at all. Okay. No, these were good questions. I never have a problem with questions. Never, ever. Again, the only bad question is the question not asked. All righty. So we put it all together. And again, we have this nice, again, here's that lateral ventricle, interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle, out the apertures or down the central canal or something into the subarachnoid space, where it floats around until eventually it goes through that arachnoid villi back into the blood supply that it came out of. So that is it says circulation, but really these are the structures involved. Remember, as we said, all of these choroid plexus, this choroid plexus here could make a cerebral spinal fluid, and it could come straight out and then straight into the blood supply, right? So it doesn't have to pass through all these structures, but it can. So these are all spaces filled with fluid that help to provide protection for our brain. Last picture, I like this picture from your textbook with this posterior view. Notice if you look closely, you can see that they've drawn the choroid plexus for us. Here we say, like I said, those two lines of choroid plexus in this space, which is the fourth ventricle. Because again, they've removed the cerebellum, so we just see the space there. Here on top of our caudate nucleus, we see the choroid plexus that lines our lateral ventricle. And here at the top of our third ventricle, which is right here, we see the choroid plexus as well. So this picture does a nice job of showing us the choroid plexus. All right. Oh, let's do one more quick thing. Let me see if they've got it. I imagine they do. But let's check because I want to explain it because I know sometimes it can be a little confusing for people. There we go. Brain, maybe. Ventricles. Perfect. There we go. Excellent. So if we were in the classroom, one of the things that you would see is this funny little model. This model, done in gray here, is basically like the kind of model you would take if you took a brain and you poured plastic into those ventricle spaces, let it harden, and then peeled the brain away. This would be the mold that you would get from this. So notice this is a structure, this is a model, but it represents the spaces. So notice as we look at this, and certainly on the exam, as I were to point at something like this, what space would this be right here? This easily could be a question on your lab exam. I could have this picture, I could have that arrow, and I would ask you, identify that space, and what would your answer be?
The choroid plexus?
No.
Or the
lateral
lateral
ventricle.
ventricle?
Right. Again, I said space, absolutely. So that the space is the lateral ventricle. However, if you look closely on this model, you will see this pink stuff that looks like someone put chewing gum on this model and left it there. That actually represents the choroid plexus. So this pink material on here is the choroid plexus. Notice instead, I could put my arrow here and ask you to identify that space. What space would that be?
The interventricular foramen.
Excellent. What space would this be?
The third ventricle.
Excellent. What space would this be?
Cerebral aqueduct.
Excellent. And then what space would this be?
The fourth ventricle.
Perfect. There you go. Excellent. Easy. See, very, very easy. This picture was on that CRC website because, like I said, they've got a lot of models that we do. This is one of the models we have in the class mod. Absolutely. All of you are excellent, excellent. All right, perfect. Okay. With that, we are done with the brain and all of its protections. So we are now to the spinal cord. We're still talking. One of the important things to remember is we are still in the central nervous system. Remember, the central nervous system is the brain and the spinal cord. So we cannot forget about that. That is important from a functional standpoint, because while obviously we think of the spinal cord as this major highway where information goes to and from the brain, right? So it's this highway of things going upwards toward the brain and down away from the brain, but it's still part of the central nervous system, and its job is to process information. It receives information, it counts beans, positive inputs and negative inputs, and then makes decisions based on that. And those decisions that it makes, right, isn't going to decide who you fall in love with, isn't going to decide where you go to college and what you're going to be when you grow up, but there are these important processes that we call our reflexes. So that when you touch something hot, you pull away from it. Or when your doctor hits you in the knee with your hammer, right, your leg kicks out, things along those lines. These are our reflexes, processing of information that takes place in our spinal cord. Makes it sound super important. And I always think of it as this big, huge, massive highway carrying information from one direction to another. But if you actually looked at a spinal cord, held one in your hand, it wouldn't actually be very impressive. It's only about 16 to 18 inches long. In fact, your spinal cord stopped growing in length at 4 years of age. Did you stop growing in length at 4 years of age?
No.
No.
No, all of you are taller now than you were when you were 4. So because of that, your spinal cord doesn't actually go the entire length of your vertebral column. Your spinal cord actually stops right about the L1 or L2 region of your spinal cord. And it's just axons that travel down beneath that. It is this tube of nerves, but one of the interesting things about it, and I'll erase my images here so I can redraw something. Here in the cervical area, it gets a little bit bigger, an area known as the cervical enlargement. Here in the lumbar region, it gets a little bit larger as well. Why might these two areas of your spinal cord be bigger?
Is it because they help control our arms and legs?
Exactly. You have more axons, you have more neurons, you have more information going to and from your arms and your legs than you do, for instance, the core of your body. So yeah, the cervical enlargement are where you have more axons to and from the arms, and the same thing for the lumbar. More axons going to and from the legs. Exactly. Simple, simple. Now, as I mentioned, the spinal cord doesn't go all the way to the end of the vertebral column, but there are axons that do that. So if we take a closer look at the inferior part of the spinal cord, we can see it's somewhat specialized. Notice the first thing I want to point out to you is where it ends. So let's go ahead and do that. Notice as the spinal cord comes down, that doesn't stand out enough. So let's do that, and let's do that. As the spinal cord comes down, it doesn't just terminate abruptly, it tapers into this peaked, or dare I say, cone-like structure. And that cone-like structure where it terminates is called the conus medullaris. And like we said, notice that's at about the L1 or L2 region of the spinal cord. But axons coming off of the spinal cord continue to travel down the spinal cord before they finally leave out those friends that we talked about before, the spaces between the vertebrae, the intervertebral foramen. And so these long drapes of axons that go either out the sacral foramina or out the intervertebral foramina form this long, straight, linear, course-like structure that is known as the caudae equina, because it looks like a horse's tail. However, as we have also mentioned, we know that our nervous tissue, like the brain and like the spinal cord, have that shrink wrap, that pia matter that is going to be on the outer surface. At the end of the cona medullaris, that pia matter, the fibers of the pia matter, travel all the way down and actually attach to the coccyx in a thread-like structure that helps to stabilize the spinal cord in place, forming a structure known as the filum terminalis. Remember, we talked about those growth spurts. This is a collagen fiber and it doesn't stretch very well. So if a child is growing very rapidly, as their body gets longer and this fiber isn't, that fiber can actually start pulling down on the spinal cord. And as you pull down on the spinal cord, that can start to cut off some of the nerves. They can get tremendous pain in their limbs, weakness in their limbs, tingling sensations as a result of this, loss of sensations from the pinching of those nerves, right? And it can be a very, very serious problem. And does anybody know how they fix it?
Do they snip it? Like,
yep.
So it's
That's
not
all
so
they have to do. They go in and cut that fiber. That allows the spinal cord to go back to its normal position, and the problem goes away almost instantaneously. Now, does that mean that your spinal cord is going to be flopping around inside of your vertebral foramen as a result of that? No, because there are other protections that are going to help to stabilize it and hold it in place. Is it a tiny bit less stable? Maybe, but is that better than having nerve damage and weakness and pain in your appendages? Yes, absolutely. In many instances, yeah, the relief is almost instantaneous. Absolutely. So yeah, it can be almost instantaneous. Now, again, it depends on how long and how severe the damage is. It can, for some people, take a little longer to recover, but most of the resolution and most of the symptoms happen almost immediately.
So I have a little bit of experience with this.
Oh, wow.
Yeah. I have herniated L3 through S1 with all of that stuff. And one of the things that they do is called an ablation. So they'll do a nerve study where they put a needle and then they put a needle right up against where they think the pain is happening. And then they pretty much attach electrodes to it and destroy it. And they use, I forget the imaging that they do, because there's imaging where they need to be able to locate it.
Yeah.
But it works.
Yeah.
Yeah, it works really
So,
good.
yeah, absolutely. So they can destroy that fiber. It helps to release it. And then also, yeah, with that pain damage, they can be localized where they can deal with that as well. Absolutely. All right, spectacular. So these are the kinds of specializations we see down at the bottom of the spinal cord. Let's talk about the spinal cord in its entirety. Also talking about the spinal nerves that come off of it. The spinal cord, as you can see from the numbering here, the spinal cord is actually divided. Oops, let's go here. Is divided up into 31 distinct regions. So that's a little big. So let's go back. Do that. So there is each one of these individual segments of the spinal cord has axons coming off of that. And that is what we call a spinal cord segment. So there are 31 spinal cord segments. And each of those spinal cord segments have axons that travel into and out of the spinal cord. And when they travel into and out of the spinal cord, they form the spinal nerves. So on the left side, you have 31. On the right side, you have 31. So you have 31 pairs of spinal nerves. All right. Oops, I didn't want to erase that. Let's go back. Now, they are named and numbered by the vertebrae. And notice if we cheat here and look, we can see, we know we have 5 lumbar vertebrae and we have 5 lumbar nerves. We have, remember, our sacrum was made up of 5 vertebrae that fused together. And sure enough, we have 5 sacral nerves. We have our 1 coccyx with its 1 coccyx nerve, 12 thoracic vertebrae with 12 thoracic nerves. And how many cervical vertebrae did we have?
7.
7. But notice here we have an extra 8th nerve. And if you look at this, you can actually see why. Because what actually happens is the first nerve actually goes over the top of the cervical vertebrae 1, and then all the rest go underneath. So they fit 1 extra cervical nerve in there. We have 8 pairs of cervical nerves, but all the rest fall in line with the number of bones that we have. All right. Now, if we, and so here's another pretty picture that now shows that. Notice how C1 goes over the top and then all the rest go down. And notice because we have to travel further and further, again, these axons start to form that cauda equina before they're finally able to leave and exit out the vertebral column. All righty. Questions on this? Now, notice as I mentioned, all spinal nerves have axons that carry information into the spinal cord, and all have axons that carry information out of So functionally, how would we identify an axon that carried information into the spinal cord?
That'd be motor.
If it's carrying it into the spinal cord, what kind of information comes inward?
Probably
Sensory.
sensory.
Sensory, excellent. So this is into the central nervous system. And what was the directional term we used for that? Like an arrow going
Afferent.
into afferent. Excellent. Excellent. Motor, on the other hand, would be going out of the central nervous system. And how did we refer to that direction? Efferent. Excellent. And I know I'm pronouncing them wrong, but again, saying afferent and efferent helps me to remember which letter goes with which. So this is what we call a mixed nerve, where it has both sensory and motor information. So all 31 of our spinal nerves are mixed. When we get to our 12 cranial nerves, we will learn that they are not all mixed. Some are sensory, some are motor, and some are mixed. So we'll actually have to differentiate those. But we don't have to worry about that with our spinal cord. It also means if we were to cut one of these nerves, right, you would both lose, right? If I cut the nerve going to this finger, I would no longer be able to move this finger, and I would no longer be able to touch with this finger. I would lose both the sensory and the motor to that finger, right? So if I cut that nerve, I lose both sensory and motor. All righty. We keep talking about these nerves, but I want to briefly, very briefly, talk about the anatomy of them. Because notice, we know a nerve is a bundle of axons. And we know axons are long thread-like materials. You could even almost say that they were kind of like fibers. Didn't we have something else that was long and thin and that we called fibers?
Muscle fibers.
Muscle cells, muscle fibers, absolutely. And so notice, just like in muscles, we have axons. Notice these axons have their myelin sheath around them. But notice this axon also has a layer of areolar connective tissue that wraps around it. Yes, Yulia.
Yes, I have a question. So when I think of nerves being highly mitotic and using a ton of energy, for some reason in my mind, it means they should also be highly vascularized. Are they at all vascular? Do they have a blood supply at all or not really?
Yeah, notice there are red and blue things here in my bundle, so absolutely there are blood vessels there. Yep, absolutely.
And so that leads me to ask, is the reason why they don't heal well because they just don't ever reproduce? Like you just kind of have them formed and the ones that die? Or like,
Well, okay, but let's be careful about that. When we talk about the death of a neuron, it is typically the cell body that dies, right? If a nerve gets cut, if you cut the nerve going to your finger, right, the tip of your finger or something like that, you may lose the sense of feeling for that. Or when I had my knee surgery, I've had 3 knee surgeries. When I had my second knee surgery, it was a 6.5-hour surgery, a tremendous amount of reconstruction that went on in there. And after that surgery, there was about a portion of my skin a little bit larger than a silver dollar where I had no sense of touch at all. Of course, that surgery was 25 years ago now. And what about that space on my knee? Do I still have no feeling there?
I'm gonna guess you gained some feeling back.
Yeah, the feeling eventually came back. It took years, but it came back. If a nerve is cut, if the axons are cut, but if an axon is damaged, but the myelin cells, the Schwann cells are still intact, they can help in the regrowth of that nerve. Now, is it always going to grow back? No, but is it possible to have it grow back? Yes. It may take years, but it can grow back. However, when the cell bodies, when the gray matter is damaged, those cell bodies aren't growing back. But yeah, often when you have surgery and some of these nerves are cut, they will grow back. So you may lose some sensation for a period of time, but typically, and again, it takes a while, but typically it will grow back. All right. All right, let's see if any of this organization sounds familiar. We have a layer of areolar connective tissue, and that areolar connective tissue insulates each individual axon. Many axons are bundled together into a structure. And just for the heck of it, we'll do that with a dense irregular connective tissue. Exactly. Let's see if we can finish that off. Notice then all of those bundles plus, as was mentioned, our blood vessels are bundled together with a dense irregular connective tissue to form the nerve. So as you just pointed out, what does this organization sound a whole heck of a lot like?
Like muscle.
Skeletal muscle tissue.
Yeah. Now, in our skeletal muscle, what did we call the areolar connective tissue that wrapped around each individual muscle cell?
Was that the endomysium?
Endomysium. Endo means within. Mesium refers to muscle. So here we can still use endo, but it's not muscle anymore. So endoneurium. What do you think we call a bundle of axons? What did we call the bundle in muscle cells?
Mesium.
Well, no, that's what paramecium is what formed the bundle, but what did we actually call the bundle?
Fascicles?
Fascicle?
Fascicle. Absolutely. The fascicle. Perfect. But you are right. It is formed by that dense irregular connective tissue that we called a perimecium. So this one's going to be the perineurium. And the dense irregular connective tissue that wrapped the whole muscle together, we called what?
Epimysium?
There you go. So the dense irregular connective tissue that bundles the blood vessels and all the fascicles up to form a nerve is the epineurium. There you go. Just that easily, you have learned the organization of a spinal nerve. It's identical to what we saw in a muscle. All right.
Wasn't there like a ganglion that was a bundle of axons, or am I just
Ganglion,
making that
remember,
up?
is a bundle of cell bodies.
Okay, thank you.
Yep. Excellent. So perfect. So again, endoneurium, perineurium, epineurium, right? Exact same forming the fascicle, exact same kind of organization we saw in muscle cells, we see here in our spinal nerve. All right. All right. And again, here's another simple illustration showing that as well. Perfect. All right. So now, before we finally get to the anatomy of the spinal cord, which we need to do, and this is going to help you tremendously on that neural review we need to talk about. The last thing I want to talk about are the protections. Again, we are still going to have those same meninges and in the same order. So we are still going to have a dura mater. We are still going to have an actually, let's do it this way. We're still going to have an arachnoid matter. And I did it again. And we're still going to have a pia mater. That all is going to be the same. And they all serve the same basic function. Our pia mater, again, is the shrink wrap directly on the tissue for protection and to stabilize the blood vessels. So that is absolutely going to be the same. Right? And we see that here with our pretty picture with that shrink wrap pia mater around there. Now, above the pia mater is again going to be our subarachnoid space. And that subarachnoid space is going to again contain the cerebrospinal fluid. And of course, that subarachnoid space is below the arachnoid matter. And we'll pretend I spelled arachnoid matter there correctly. So notice this beginning part of it is all the same and all identical to what we saw in the brain. But here's where things get a little different, starting first with the subdural space. Remember, in our brain or in the cranial cavity, that subdural space is a potential space with nothing inside of it. But here in the spinal cord, it is an actual space. And this actual space contains interstitial fluid. Now, of course, the subdural space is between the dura mater and the arachnoid mater. And someone earlier to me mentioned an epidural space, something we don't have in the skull. But is here in the vertebral column. It is a space between the bone and the dura. And it contains adipose and areolar connective tissue. And there you go, absolutely. Just real quickly to finish things off. Obviously above the epidural space is the bone of the vertebrae. And it has a periosteum. So notice here in the vertebral column, the periosteum is not a part of the dura mater. I would ask you why this is the case, but we've already talked about it and we've already explained it. The whole reason for all of this is that our vertebral column needs to be able to move and our bones of our skull don't. So notice, as we see here, we have the bone, we have the dura mater, and the epidural space containing the adipose and the areolar connective tissue. And yes, where most people think of the epidural space is when you are giving birth. One of the big questions people ask you is, do you want an epidural? Well, an epidural is where they take a big needle and they inject an anesthetic into this space, the epidural space, where that anesthetic can then diffuse across to the spinal cord and paralyze those nerves so that you don't feel the pain as much. Of course, it also paralyzes the motor one, so you can't walk around anymore either. You're bedridden as a result of it, but you feel less pain. However, this is a very small space. And if that intern who's doing it happens to go a little too deep, they could potentially jab the spinal cord. So how do they resolve that?
They only
Do
have
they do?
highly trained technicians do it.
That's adorable. Now, how do
Do
they?
you give it in the lumbar spine when the
And
oh my
not
gosh,
just
where the
the
spinal
lumbar,
column
but
ends?
exactly. Notice typically where an epidural is given is either somewhere around the L3 to L5 region. Notice in that portion when they're going into that epidural space, that is below the end of the spinal cord. So this is just where those caudae equina, where those roots, those axons are located. So you're much less likely to damage the spinal cord, right? Because then just the nerves there. Now, pushing through into the dural space, through the dura is still a problem. It can cause a leaking of cerebral spinal fluid. That can cause a drop in pressure. It can cause terrible migraines, some other minor pressures that can occur as a result of that leaking of cerebral spinal fluid as well. So it's still a concern if they go too deep, but by doing it much, much lower in the back, they are certain they're not going to hit the spinal cord.
What about sometimes people will only be numb on like one side or they'll rotate you? Is that just a placement issue?
Exactly. Yeah. If we go back to this picture, much like the brain, the spinal cord has two hemispheres, hemisphere 1 and hemisphere 2. So if the injection is too lateral, then it's much more likely to just inhibit the nerves on one side. And so you can get an asymmetrical effect. So yeah, one of the things they'll try to do is try to rotate you to try to increase the circulation and movement of that to get it bilaterally. Yep, the goal is to get it as close to the middle as possible so it'll evenly diffuse to all the nerves. All right, so again, we did from the inside out, but again, well, and again, that's one of the reasons why it's not a required thing for childbirth, but the risks associated with it are relatively minor. And so it's that weighing of the decision between the amount of pain that occurs during childbirth versus the comfort of it. There is no wrong answer. Some people are very comfortable have it. Some people don't want it. Some people, because of the time that it takes for it to kick in, if you're too close to giving birth, it won't have time to kick in, so you may not have the option of getting it. There are other things like that, but again, so those are all more philosophical. We can talk the anatomy of it, but I'm not going to discuss. Yeah, if it's an emergency surgery, then that's something totally different as well. All right, so again, here we identified all these layers, talked about all of them from the outside in epidural space, fat and connective tissue, then the dura mater, then the subdural actual space with interstitial fluid, arachnoid matter, subarachnoid space, and then the shrink wrap pia mater. All right, those are the protections. How are we on time? Yikes. So let's talk now about the anatomy of our spinal cord. And I'm going to draw this. If we look at our spinal cord, it is an oval shaped structure, which as we know is going to have a tiny little hole at the center. And that tiny little hole in the center is, of course, our central canal, containing cerebral spinal fluid. Ooh, and let's actually make this a little bigger. I'll cheat. So I have some room to write in it. This is indeed our central canal. But one of the cool things about our central canal is it is a fluid-filled space, and this fluid-filled space is going to be lined by a very thin layer of cells. Any idea what cells may line the inner surface of this central canal? Well, it's a fluid-filled space, so it needs like an epithelial tissue. If only we had a neuroglial epithelial tissue we were able to take advantage of there. What would something like that be?
Are they ependymal cells?
Ependymal cells, absolutely. This is another location where you will find ependymal cells. One of the interesting things that happens is as we age, the number of ependymal cells actually increase in the central canal, and it can actually congest it almost entirely so that it blocks the flow of cerebral spinal fluid through it. Doesn't have a major impact on function because remember, it's still going to bathe the outer surface, but it's one of the indicators of age when you look at these things histologically. What's interesting about the spinal cord is unlike our cerebrum, unlike our cerebellum, in the spinal cord, the gray matter is deep and the white matter is superficial. There are 3 enlargements of the gray matter. One of them is to the back, one of them is to the side, and one of them is to the front of the spinal cord. And this is supposed to be symmetrical, so pretend that's symmetrical. These 3 enlargements are what are known as the horns. One horn is towards the front. It is the biggest horn, so it is the anterior gray horn, and it is the largest. It's one of the ways you can tell the front and the back of our spinal cord. That means the one to the back, of course, is going to be the posterior gray horn. And because I use the terms anterior and posterior here, what other terms could I have used interchangeably with it? Anywhere I write posterior, what else could I write?
Dorsal.
And anywhere I write anterior, what could I write interchangeably?
Ventral.
Ventral. Absolutely. And it will be correct either way. Doesn't matter which one of those 2 you use, you're welcome to use them either way. So anterior gray horn, posterior gray horn, and then of course the one in the middle would be middle, but it's also more to the side, so it is the lateral gray horn. Our white matter is to the outside, and it is also divided into what we call the white columns. So over here is the lateral white column. Back here is the posterior white column. And up here is the anterior. Yes.
So a good way to know our orientation is to look for anterior gray horn, and it would be facing like our guts.
I'm sorry, say it again.
Would it be facing like our abdomen and our guts and heart and lungs?
Okay. In the body, yes.
Okay.
But if I put this on a microscope slide, does it matter which way I put the slide on the microscope?
No.
No. So on the microscope, you have to be able to tell the 2 sides apart. And so luckily, there's a couple ways to do that. The first is that the anterior gray horn is the largest. So that's one way you can tell the front from the back. Also, remember I mentioned that these are divided into hemispheres, a right and a left hemisphere. And so not surprisingly, there is an invagination on the midline in the back that is known as the posterior, because it's in the back. Median, because it's on the midline, and a folded invagination sulcus. So I have a posterior median sulcus. However, we have something similar in the front, but in the front, it's much, much deeper. So based on that, on the front, it's going to be an anterior median. And since it's a really, really deep sulcus, what would we call it?
Fissure?
A fissure. Excellent. And there is a third way we can tell the front from the back. Remember, each segment of the spinal cord has axons coming into it and out of it. Some of those axons are connected to the back. Some of those axons are connected to the front. The axons coming off, oh, let's do it on the other side too. Since I'm labeling this side, this will work. Totally cheat here. There. There. Oops. We know the axons come together to form our spinal nerve, which of course, as remember, we know we have 31 pairs of. And some of the axons come off the back, some of the axons come off the front. So the ones that come off the back, we call the posterior or dorsal root. And the ones that come off the front are the anterior root. Now, the roots themselves don't really tell us a whole heck of a lot. However, on our dorsal root,
Would posterior root also be okay there?
posterior root would also be acceptable here. Absolutely. Again, the words are completely interchangeable. On the posterior or dorsal root, uh-oh, didn't want that. All right, screw it. I'll just fix it. Is a big, large cluster. And this big, large cluster is a collection of cell bodies. And as we know, this is just getting out of hand. There we go. A collection of cell bodies outside the central nervous system is a ganglion. So this structure is the dorsal root ganglion. Notice there's no ventral root ganglion or anterior root ganglion. So whichever one the ganglion is on, that is the one where there is the, so fix that. Excellent. And then come back here, that. Put a big ganglion there as well. Excellent. So whichever one has the ganglion is the posterior side. So those are the three ways that you can tell which is which. Anterior gray horn is the largest horn. Anterior median fissure is the biggest fissure. Dorsal root ganglion is a big cluster on the posterior side. So if you use those three things, no matter how this is oriented on a slide, you should be able to tell what is what. All right.
Did you tell us what the dorsal root ganglion holds?
I haven't. Well, it's a ganglion, so we know it holds cell bodies. But we also know that the gray matter holds cell bodies as well. So that's absolutely where we're going next. Because notice what I've given you here are a whole lot of locations. And especially when we're talking about gray matter locations, that gray matter locations are going to contain cell bodies. And those cell bodies in those locations are going to have a specific function. So let's talk about that. As I mentioned, the anterior gray horn is the largest. So in here are some cell bodies in the anterior gray horn. their axons leave out of the anterior ventral root, become part of the spinal nerve, and they go to skeletal muscle. Based on that, what is the function of this anterior gray horn cell?
Motor.
And you'd get partial credit for that, but can we be more specific?
Sensory motor?
Close. Somatic, absolutely. Somatic motor. Remember, somatic motor is what controls skeletal muscle. And now we have a location. Now we have a function. Do we know the structural classification of a neuron that is a somatic motor neuron?
Bipolar.
What was the structural?
Would they be multipolar?
Multipolar. Excellent. So its structural is going to be multipolar, which also means we have to come here and give it some ganglia. I mean, give it some dendrites. Perfect. Excellent. All right. Now, as someone mentioned earlier, up here in our dorsal root ganglion, we have some cell bodies, because ganglia are a collection of cell bodies. As it turns out, this particular cell body has a single peripheral axon to it. Its job is to carry information in to the spinal cord. So for instance, this is touch or, tickle, pain, right? This is our sensory information, which is being carried afferently in. So based on this, what is our functional classification of this particular neuron found in the dorsal root ganglion?
Sensory.
And because I've drawn it and because we know what most sensory are, what is the structural classification of this neuron going to be?
Multipolar.
Unipolar. Excellent. So once again, we have a location, dorsal root ganglion. We have a function, sensory. We have a structure, unipolar. That leaves us with our lateral gray horn. Notice I made a big deal with our, saying somatic motor with our lateral gray horn, with our anterior gray horn, because while it connects to skeletal muscle, we need something to be able to control, to go to our smooth muscle, our cardiac muscle, and our glands. And what type of neuron would control our smooth muscle, our cardiac muscle, and our glands?
Wouldn't it be, I want to say interneurons, but that doesn't make sense. Never mind.
What goes to smooth muscle, cardiac muscle, and glands? It's clearly not somatic motor, but it is motor. What other kind of motor did we have?
Bipolar?
Say again?
Bipolar?
No, we're talking function here.
Unipolar?
You're still talking function here. You're giving me structural classifications. I'm asking for functional classifications. It's not interneuron. It's not sensory. There we go. Autonomic motor. So this is going to be an autonomic motor neuron. Now, if it's a motor neuron, what must its structural classification be?
Multipolar.
There
Multipolar.
you go. Multipolar, excellent. So let's give it some dendrites. Now, when it comes to its axon, where do you think its axon's going to want to go? Is it going to want to go up here with these sensory axons, or do you think it's going to want to go down here with the motor ones?
I think with the motor
Sensory.
one.
Yeah, it's going to go with the motor. Like likes like. So it's also going to go to the anterior root, come out to the spinal nerve, and go to our cardiac muscle, our smooth muscle, and our glands. So notice when we think of these things from a functional standpoint, our dorsal root contains sensory axons only. Our anterior root contains motor axons only. But remember, as we talked about, our spinal nerve, we talked about being that mixed nerve, and it contains both sensory and motor axons.
And these are symmetrical on both sides, so those neurons are the same where you have the writing.
Exactly. The only reason I've separated it is so that I could write the words on one side and draw the pictures on the other. But absolutely,
Yeah.
completely symmetrical.
It actually makes it really easy to see like this. I was just making sure I had the right idea.
That's because I'm an amazing instructor.
Touche.
No, thank you. I appreciate that. Yeah, no, I separating it helps me as well. So I think it's useful that way. So, questions on that? Excellent. So again, notice one of the key things we've been emphasizing here is I've given you a bunch of structures or bunch of locations, right? And for each of these locations, the neurons that are associated with it, we know their function. And if we know their function, we know their structural classification. So structure, function, location, all go hand in hand in hand. And while we've done this drawing here, which I'll save before I forget. Yes, go ahead, question.
Yeah, I was trying to do the nervous system review last night, and even like as we're covering this, I'm not sure what it means. Question 9 says, what structure is formed by the axons of a neuron in the anterior grain horn when they leave the spinal cord? So are you looking for a structure as in like a ganglion or what?
Well, seeing as this one is graded for correctness, I'm not sure I could answer that. But since we are here collectively as a group, ask us all the question again.
So you're looking for a structure that is formed by the axons of neurons in the anterior great horn when they leave
Okay,
the
so
spinal
stop.
cord.
So excellent. So using my highlighter, and we'll do this on the left side to see if we can make sense of it. We are talking about a neuron that would be found here in the anterior gray horn, correct? Is that what you told me?
Yes. Mhm.
Excellent. So where does its axon go?
It goes out to the, well, down under through the anterior root and then into the middle.
Okay, eventually it ends up in the spinal nerve, but what's the first structure it helps to form?
Or would it be like anterior root?
I don't know, you tell me. Anybody? Do you think, what do you guys think the answer is?
I guess if they're always there, then it would be part of the structure of the anterior root.
There you go.
I feel like it's hard sometimes to differentiate, like if something's just running through it or if it's part of it.
Well, so again, it's easier when we're on campus because I can use the analogies there. Auburn Boulevard, there is blacktop that is there that forms Auburn Boulevard, right? So the road is made from the blacktop. What is the anterior root made of?
These neurons.
Yeah.
or the axons of these nerves?
Axons. They're made of axons, absolutely.
So if they're made of axons, is it the bodies or the cell bodies that lie in the gray matter itself, or
Yes.
what?
Remember, gray matter is the cell bodies and the dendrites. White matter is the axons. Nerves are a bundle of axons, right? We just looked at the organization of a nerve, right? Those bundles of axons, that's what this is. This anterior root, the spinal nerve are bundles of axons. So it's made of axons.
Okay, but with the dorsal root ganglions, their bodies lie in that ganglion they form at
Right.
the dorsal side.
Notice that's not a nerve. That's a ganglion. Ganglion are gray matter. Gray matter have cell bodies.
Okay, it's slowly starting to come together.
Yep.
And the fact that they're unipolar probably also plays a role in.
Well, that, yeah, it has to do with their function, because their function. But yes, the short answer to anything that is made of white matter is made of axons, primarily myelinated axons. Right? That's why it's white, is because the fat of that myelinated axon. Whereas the gray matter is the cell bodies that do not have white matter on it. So when we look at this tissue, we have white matter and we have gray matter. And again, I think I closed it again, but let's open it back up.
Is the white or the anterior root considered white matter, or is it not myelinated at that point?
Say that again.
Is the like anterior root or the dorsal root considered white matter, or is there no myelination at that point?
No, it's
of the
still, yeah,
axon?
no, the axons, yes, they're still, yes, the roots are still made up of axons, right? So yes, so they're still white matter. Right. That doesn't show it. Do I have a picture that shows it somewhere? Right. Here's an actual histology side of the spinal cord. Where's anterior, up or down?
Down.
Exactly, right? And as we talked about, there are three key ways that we can tell that. One is by the largest the largest horn being anterior, right? The largest fissure being anterior. And then notice our root coming off connects to and has that big ganglion on it. Right. So notice the ganglion is kind of towards the front, but if you follow the root, you can see the ganglion connects to that dorsal root. So this is the back, this is the front. Right. What about this one? Notice this one, we don't see the dorsal root ganglion, but again, we can see that nice big anterior root, that nice big ventral fissure, median or anterior median fissure, right? And the fact that it's labeled kind of helps with this one as well, right? So we see all of those. And then here's another one where we can see, again, that larger ventral root, which is usually too obvious. This is a crappy picture that's kind of fuzzy and out of whack. Oh, there's some ependymal cells around our central canal, some of the histology we were looking at. And oops, we should actually go back to this one. These cells here, this is the anterior gray horn. These are our multipolar somatic motor neurons found in the anterior gray horn. So yeah, so again, location, function, structure, all go hand in hand on these things. All right, so let's go ahead back to the lecture and look at the pretty pictures from there. Again, we've talked about our white matter and our gray matter with those three horns, anterior, posterior, and lateral. Here we see another nice illustration. And again, largest is the anterior gray horn with our somatic motor. Then we have our lateral gray horn with the visceral or autonomic motor. And then our sensory, all sensory is located up here in the posterior gray horn. Here's our dorsal root ganglion, which has those unipolar neurons carrying information in. And then here in these areas, we have our multipolar going out the anterior root to the spinal nerve. Yes, question.
Yes, the book labels the white columns in like 3 extra like subdivisions. Is that beneficial to know, or is it just
Yes, so notice there, like we talked about and I drew on the illustration, we have our posterior white column, our lateral white column, and our anterior white column. And as precisely organized as the gray matter is, the white matter is just as organized. Notice here, we see this great illustration from your textbook that actually shows the individual tracks or actually different type of information. Some of it is sensory, going up towards the brain. So we call that ascending. Descending is motor going down. So absolutely, it is just, the white matter is just as precisely organized. I'm just not holding you responsible for this, right? We could literally spend a whole semester just talking about the nervous system. There's so much complexity to all of this. We're obviously not going into that depth, but I did want to at least show you that even though we're not talking about it, the white matter is just as precisely organized as the gray matter is. Where some areas are sensory, some areas are motor, carrying very specific types of information. So, but all you need to know is the 3 regions: the posterior white column, the lateral white column, and the anterior white column. You don't need to know all of the individual tracks that are in it. Okay. We did that, we did that, we did that. Actually, we didn't do that. All right, questions on this?
I just have
Yes.
clarification.
Yeah.
Sorry. So the lateral gray horn is responsible for things that are not like conscious decisions. They're like breathing, heart rate, stuff like that.
No, the lateral gray horn, again, motor areas control the motor effectors. Those are muscles and glands. So there are axons that come out of the lateral gray horn that go to your heart to control your heart rate, that go to your stomach to control your rate of digestion, to do all those types of things, right? So they are the motor output that tells your smooth muscle, your cardiac muscle, and your glands what to do. And yes, you are right. Most of that stuff is involuntary. It's outside of our conscious control. But you got to be careful when you say things like breathing, because breathing is controlling skeletal muscle to move air in and out. That is a reflex, but that's not controlled by the lateral gray horn.
Okay, so they have motor autonomic neurons.
Correct.
Okay. Oh
So
my
those
God.
are neurons whose axons travel out of the central nervous system and go to smooth muscle, cardiac muscle, and glands, those effectors to tell them what to do. So whether you're sweating, whether you're salivating, whether you're crying, right? Whether your stomach is churning, your heart is beating faster, any of those types of things. Arthur, were you the other one that I saw who had a question?
Yeah. Let me scroll down to it. I don't know if I might have just missed it. Dorsal Ramus and Ventral Ramus.
I was starting to go there, and then I realized we hadn't done
okay.
that yet. So you're right. So that is the last thing that I think we need to talk about for this for now. Where are we on time? Yowzers. Okay. So I really haven't given myself a lot of room to do this. Okay. So I'm going to cheat and redraw some of this. Oops. So here's my anterior root containing the motor only. Here is my dorsal root containing sensory only, and my dorsal root ganglion. And this comes together to my spinal nerve. So this goes there. This goes here. This goes there. Excellent. Whoops. Ah. All right, good enough. So we know that these two roots come together to form our spinal nerve. But our spinal nerve, well, it's easier when I have the whiteboard and I can erase these things more easily. So you don't just have 31 nerves that go to 31 locations in your body. Your spinal nerve, almost immediately after it goes out the intervertebral foramen, branches again, not once, not twice, but actually 4 different branches that come off of that spinal nerve. The largest of these branches is the ventral ramus. The ventral ramus goes to the anterior part of the trunk, and it goes to the and the limbs, so the arms and the legs. So that is the largest branch, the ventral ramus. Next is the dorsal ramus. This one goes to the skin and muscles of the back. And then these other 2 rami together are what are known as the rami, because rami is the plural of ramus, communicantes, which is incredibly fun to say. But we also want to identify them individually. The one that is more medial is what is known as the gray ramus. And the one that is more lateral is the white ramus. So we have these 2 rami communicantes, the lateral white ramus, the medial gray ramus. And these are involved in these are pathways of our sympathetic nervous system. Right? So basically, these are pathways to our internal organs.
Is the gray and white label, the gray ramus doesn't have like cell bodies or anything. It's just mostly like more deep. One's deep and one's superior, kind of.
One's medial and one's lateral. The reason for their names we will get to, but we'll get to that when we get to the autonomic nervous system. For right now, you just have to know the names. But next week, we'll start talking about why they're called what they're called. But we don't have to worry about that.
Thank you.
We'll get there though. All right, excellent. So notice if we go back here, and again, we have the ventral or anterior and dorsal posterior rami. Here we have this great picture that does a really nice job of showing this. Notice here we have our spinal nerve that branches into the dorsal ramus, which, as I mentioned, goes to the skin and muscles of the back. We have the ventral ramus, which here in the trunk goes to the anterior part of the trunk. But if there was an arm here or a leg here, it would also go to that arm and that leg. And then really tiny right here, you can see the white ramus and the gray ramus, those rami communicantes, which again, sounds like a fun drink that you would have at a Mexican restaurant. I'd like a large rami communicante, please. Which collectively are known as those, but are, again, the lateral one is the white, the medial one is the gray ramus. And I think I have one other picture that shows this as well. Here we go. Excellent. Notice again, great picture. Here's our dorsal root ganglion. Here's our ventral root, spinal nerve, and then our dorsal ramus, our ventral ramus, our white ramus, and our gray ramus. Those 4 branches off of that spinal nerve. And again, those rami communicantes, the gray and white ramus, are part of our sympathetic pathways, which we'll talk about next week. All right, excellent. Questions on that? All right, so here's the good news. We have gotten through all the lecture that I wanted, but I had really hoped to get through the cranial nerves today. We are not going to be able to get through the cranial nerves today, but I think it's still worthwhile to start them, because if you understand how to do the first few, then over the weekend you can kind of work on the other ones yourselves, and then we'll come back together and work on it together at the beginning of class on Thursday. What day is today? Today Tuesday? Oh yeah, today's Tuesday. Okay, excellent. Thursday. Okay, excellent, perfect. All right, even better. I'm all screwed up. Okay, excellent. So we will start it today so you can see how the process is, and then we will finish it at the beginning of the class on Thursday. All right, questions on any of that?
I have a question.
Yes.
So am I mistaken, or did we cover everything in our study handout that you gave called Gross Anatomy of the Nervous System?
That was the goal. The goal of this was to finish that list off. So I believe
Okay.
we have covered everything on it. Yes.
That's what I thought. Thank you.
That was my goal. That was my hope. So yes, in talking about this, yes, we've also done all of the anatomy. All right, excellent. So, like I said, between that and the histology, that's going to be about 50% of your lab exam. So what we need to start next is our cranial nerves. So let's start on those. Let's go ahead and take a break. Let's go ahead and we'll take a 13-minute break so that we're not too far behind, and start at 3:40. So let's do this. I will stop sharing that for a second so I can close that. And then close that. Which brings us here. So we will take a break, and starting at 3:40. So if you don't have your handout printed out already, now's the time to do it, or at least get some graph paper or some paper that you draw some lines on to help you to go through it. And we will talk about that handout and what you can do to help you to be successful on that. All right. Questions on any of that? All of them. All right, excellent. I will see you guys in 15 minutes or 12 or whatever it is. All righty. So let's switch gears to our cranial nerves. Save this one last time to make sure I have it so I can clear it. If you notice on your cranial nerves handout, there are 5 pieces of information on there, but really 4 pieces of information. You'll be tested on. The first and most obvious of those should be the name and number. Oops, why didn't that write? Of the cranial nerve. The second piece of information that you're going to be responsible for is the functional type. Remember, we just finished talking about how for our spinal nerves, all of our spinal nerves are mixed nerves, where they're a combination of both sensory and motor. But I also told you that that wouldn't be the case for cranial nerves. For cranial nerves, there are basically three functional types. There are some that are going to be sensory only. There are some, actually, let's do it this way. Do functional type there, and then I'll do the smaller underneath it. Some that will be sensory only. Some that are motor only. And some that are mixed, or the term we can also use for that is both, in that they have both sensory and motor. Okay. So the good part about this is, notice if I ask you on an exam to identify the functional type of a cranial nerve, how many possible answers are there for that question?
3.
3, either sensory, motor, or both. Now, knowing its functional type is going to be important, but again, we also need to know their specific function, which is going to be precisely what it is that they do. And the last piece of testable information is to remember that these are cranial nerves. By definition, these are bundles of axons that come off of our brain. All right, comes off the brain. They're cranial nerves, so the brain and brain stem, and go to somewhere in the body. Now, to get to somewhere in the body, they have to leave the cranial cavity. Luckily, way back at the beginning of 431, well, in the middle of 431, we learned all sorts of bones and bone features and lots of holes in that skull. And those holes in the skull are passageways for things like blood vessels and things like nerves. So the fourth possible question on all of these cranial nerves, I'll point to one of these, and I could ask you to identify its skull exit. And its skull exit is basically the bone and bone feature. Actually, let's do it this way, like we did the last one. The skull exit is going to be identified as the bone and bone feature. Or it could also be a skull feature that the nerve passes through to leave the cranial cavity. So those are the four pieces of testable information. Notice on your handout, there is a fifth piece of information. That fifth piece of information is what I call the brain exit. And the brain exit is basically just where it comes off the brain. I don't consider that a piece of testable information. I consider that what you're going to use to figure out what nerve it is. So there's no wrong answer for this. The key for you on this brain exit is for you to write something that makes sense to you, so that when you look at a picture like this, you and I on the test have an arrow that points to this right here. Well, the fact that you have a nerve that is under the pons close to the midline, right, helps you to recognize that that is the sixth cranial nerve. So again, for that brain exit, it's really, that is a piece of information for you to help to tell these things apart. All right. Now, couple tricks to all of these things, starting first over here with the names and numbers. When we're identifying the names and numbers, you are welcome to use, well, let me say it this way. It is more common to use Roman numbers. So 1, 2, 3, 4, 5, 6, 7, whoops, 8, 9, 10, 11, 12. You are welcome to use that. Let's go ahead and make that bigger and space them out a little bit more. Not going to be able to fit them all on here if I do this, am I? That's all right. We're not going to get further than that, so that'll work. So you're welcome to do it that way, but you're also welcome to use the Arabic numbers if you want, right? So you can write 1, you can write 2, and 3, and so on and so forth. So I don't care whether you use the Arabic numbers or whether you use the Roman numerals. Roman numerals is by convention what is normally used, but for this exam, as long as you give me a number, I don't care how you write it. To help you to remember the name and number, I have provided you with mnemonic 1. Oops.
Does it have anything to do with drugs?
No, although I'm sure if you do a search online, you can find plenty of those. The mnemonic for this one is, once one takes the anatomy final, very good vacations are heavenly. That is your G-rated version of this. There, you can get on the almighty internet and find all sorts of them, ranging from PG all the way up to more of the R-rated. Again, the R-rated ones are sometimes a little bit more fun to remember. I will not share the one that I learned in graduate school because it's not appropriate. But again, there are all sorts of fun ones that can help you to remember that. All right, so that is the first mnemonic. And of course, like all mnemonics, again, the first letter of the words helps you to remember the first letter of the name of it too, that you're responsible for. The second mnemonic that I've provided for you, doesn't need to be, is to help you to remember the functional types. For the functional types, that mnemonic is some, say, marry, money, but my brother says big brains matter most. So again, these are mnemonics you are welcome and encouraged to use or to find your own. And the advantage of them is right away, they help you. And let's go ahead and take that all the way across. There. There. So we'll do the first 6 because that'll be easy. Excellent. And what's nice about this is because of functional types, since there's only 3 possible answers, the functional type should be really, really simple and straightforward. What do you think the functional type of cranial nerve 1 is going to be?
Sensory.
Excellent. What's the functional type of cranial nerve 2 going to be?
Sensory.
And 3?
Motor.
And 4?
Motor.
Sensory.
Motor. And 5?
Sensory.
Both.
Both. Right? Again, oops. The letters tell you what they are. S for sensory, M for motor, B stands for both. Now again, both could also be mixed, but mixed and motor both start with M, so that's why I use both. And again, and I'm not the one who came up with this mnemonic, so again, but clearly lots of people do it this way. But that is an easy way to be able to figure this out. All right, now let's take this one step forward because this is super easy, especially for the first one, because it's one we're probably pretty comfortable with. What is the first cranial nerve?
Olfactory.
Olfactory.
Excellent. What is its functional type? Sensory, motor, or both?
Sensory.
Sensory. What is the specific function of our olfactory nerve?
Smell.
Yeah. Sense of smell, or olfaction if you want to be tricky. And how does it exit the skull to get to where it needs to be? So it can provide that sense of smell in the nasal cavity. How does it get to the nasal cavity? What is its skull exit?
Just to guess, would it be olfactory foramen of some sort?
There we go. Olfactory foramina. What were those olfactory foramina a bone feature of? Of what bone?
Ethmoid bone?
Ethmoid. And there you go. Just that simply, you have filled out the first chart, part of the chart for our first cranial nerve. It really is just that simple. So now that we know how to play the game, let's go through this some more. Your first key to identifying the cranial nerves is to identify them here on an illustration or something like that. Typically, our cranial nerves go from anterior to posterior, and when needed, typically they go medial to lateral, but that's not always the case. So notice as we work our way through it, this is cranial nerve 1 and 2 and 3 and 4 and 5, 6, 7, 8, 9, 10, 11, and 12. So notice 1 comes off the olfactory bulb, 2 is that optic nerve. Notice both 3 and 4 are above the pons. 3 is medial and 4 is lateral. 5 is the only one that's on the pons. 6, 7, 8 are below the pons. 9, 10, and 11 are along the lateral aspect of the medulla oblongata, and 12 is towards the middle. We see this on a nice illustration of the brain, and your book's got a nice picture of an actual brain where we can see all of these as well. Your book's also got a nice table that basically gives us the information we were just talking about. Notice if we look at our first one, olfactory, notice it has a sensory structure, but no motor function. So it is functional type is sensory, right? Whereas the same thing is true for our optic, sensory, but no motor function. Whereas ocular motor, no sensory, motor function. So that's motor, and so on and so forth. Notice 5 has both sensory and motor, so that's the both. So again, it does a nice job of showing us these the way, giving that specific function. And here we can look at individual nerves. Now, this is anatomy we are very comfortable with, so this is a very, very good starting point. Notice when we were talking about the limbic system, we made a strong point of emphasizing how the olfactory bulb sits here on the cribriform plate, and then the olfactory tract are the axons carrying that information back to the limbic system. I always think of this in terms of a hairbrush, right? If this was a hairbrush, this would be the handle of the hairbrush. This would be the head of the hairbrush. And then coming off of the head of the hairbrush would be all the bristles. These bristles coming off are each individual axons, but collectively they're considered the olfactory nerve. This is the one nerve that really isn't just one structure. Most nerves, when we think of them as a singular structure, a single bundle coming together, that's not true for this one. They are not in one structure. They're all these individual fibers of that brush going through all those tiny little holes, the cribriform plate, to get into the nasal cavity, so that as smells come wafting up through our nasal cavity, we perceive that sense of smell. It has the chemoreceptors that receive that information and carry it to our brain. All right. Yeah, I used the example of the boy that my sister was dating, where he actually fell down a flight of stairs, tore his olfactory bulb off of his cribriform plate, and lost his sense of smell. So damaging the olfactory bulb could be one of the ways that does it. Damaging the nerves, if you're a chronic smoker or something like that, that can cause damage to it that way. Or it could be damage inside of the insula of the limbic system where you could have that problem as well. So those are things that could occur. Yes, Laura, go ahead.
Oh yeah, what Amanda just asked. I was about to ask about the loss of smell from COVID.
Great question. We don't fully understand why that occurs. It might be possible that it damages these nerves. So there may be something about these nerves that are damaged. I don't believe they know what the cause of it is. It can be a concern because, as you notice, this cribriform plate basically is the only boundary between our olfactory system and our nervous system. So if the COVID is getting in here and affecting, causing inflammation of our olfactory bulb or in some other way stopping that sense of smell, that could lead to the potential of more severe neural trauma from that vaccine. So how it does it is not fully understood, but I know it is a concern because it could mean that hopefully it's just impacting the axons down here, maybe blocking the receptor sites or something like that. But if it's getting into the nervous tissue, then it does have the potential of having more long-term potential implications.
Is it the inflammation of the olfactory bulb or the nerves and axons?
Like I said, I don't believe, again, it isn't something I've actively looked at, but my understanding is they don't actually know the cause of the loss of smell. So I'm speculating that those could be some of the potential causes. But as far as I know, I don't believe they have determined how it causes the loss of smell. So I don't know. I don't know how that occurs. Those are ways that it could, but I don't know if it does. I don't know how it actually does it. But yeah, blocking or inflaming these nerves so that they didn't work properly would certainly be the easiest way to do it. And since it's a fairly common symptom, that would be my guess, but I don't know if it's been studied.
And also considering that it comes back
Yes.
usually means
Well,
that maybe there's not like severe.
Well, and that's the other thing. One of the truly amazing thing about these nerves in the nose, these olfactory nerves, is these are some of the very few nerves in the body that actually can regenerate. Right? In fact, they themselves only last for a couple weeks and then are constantly being replaced and regrown. When this was discovered, this was a huge finding, and massive numbers of neuroscientists went into the field of olfaction, because if we could figure out how it is that these neurons regenerate, then maybe we could use that same technique to get spinal cord nerves to regenerate so people could walk again when they've been damaged or stuff like that. But they've never been able to figure out what's so special about these neurons that allows them to regenerate, or how to translate that to another neurons. They've had much more success with embryonic stem cell research, and so a lot of the field has gone more that way than into olfaction. So not nearly as many people are studying olfaction in the nervous system than there were before. But yeah, these. So again, it is likely something with these nerves, but I don't know that for certain. All right, great question. I wish I had an answer for you, but it's an awesome question. Any other questions on this process? We understand how the game is played? Excellent. So what is cranial nerve 2?
The optic nerve.
Optic nerve. What is its functional type?
Sensory.
And what is its specific function?
Vision.
There you go. Absolutely, right? This is another one that we've spent a fair amount of time talking about. Coming out of each eyeball is a bundle of axons that we call the optic nerve. Remember, we talked about how those optic nerves come together to the optic chiasm, where some axons cross and some don't. So that side specific information goes to the thalamus, to that lateral geniculate nucleus of the thalamus, and from there goes to our primary visual cortex. So again, this is a nerve we've talked about before. We saw it cut here on the brainstem, where it comes together to that optic chiasm and comes out. And again, this is a perfect example of that last thing, the brain exit. You write what makes sense to you. It's a lot easier. Let's go back for a second. It's a lot easier here because really the olfactory nerve comes off the olfactory bulb. So its brain exit is the olfactory bulb. That's pretty simple, right? You could say the telencephalon if you want, but why make it more complicated? It is the only one that comes off the telencephalon. So it does make it unique that way. But it comes off the olfactory bulb. That's pretty straightforward. For the optic nerve, well, you could say it's the one that comes out of the eyeball. Of course, if your model doesn't have the eyeball, then you could say it's the one in front of the pituitary gland and the infundibulum, or you can say it's the one that comes off the optic chiasm, or you could say it's the one that comes out of the thalamus. Again, there's no wrong answer. You just need to write whatever makes sense to you. All right, so we've got 3 of the 4 big ones. We know it's the optic nerve, number 2. We know it's sensory. I think we mentioned the specific function, but I'll ask it anyway. What is the specific function?
Vision.
Vision. So then the last question is, how does it exit the skull to get to the eyeball? Does anybody remember?
The occipital cortex?
No, that's where it talks to the brain. But remember, if we think back to the skull, wasn't there kind of a VIP entrance that our optic nerve passed through to get to the orbit of the eye? The superior orbital fissure is definitely a way to get to the eye, but that doesn't sound like a very VIP entrance to me.
Optic foramen.
Optic foramen, absolutely. If you remember, there was that special, that doesn't show up very well. There is that doesn't show up very well either. There was that special VIP entrance, the optic canal or optic foramen, which was the pathway for the optic nerve to get to the orbit of the eye. Remember, if we looked at it from a superior view, remember one of the things we talked about is these optic canals. This one travels this way, and this one travels this way. So you can actually see how the axons are going to come together and cross to form the optic chiasm. So that nice VIP exit, the skull exit for our optic nerve is the optic canal, or optic foramen would be fine as well. All right.
Do you just want the bone feature, or do you want like the optic foramen of the?
Well, I want bone and bone feature. So you're absolutely correct. The optic canal of what bone? The optic canal is a bone feature of what bone? It's the purple one we see right here. What bone is that?
Ethmoid?
Nope.
The sphenoid?
Sphenoid, absolutely. It is the sphenoid. Excellent, excellent. It is a cumulative class, and this is information we definitely need to know. So yes, I do indeed want the bone and bone feature. Now, if it's a skull feature, obviously you don't have to worry about the bones. But if it's a bone feature, I want the bone. Okay. All right. That's 1 and 2. Those were easy. Let's get to the next ones. But before we learn the next one, we have some more work we have to do. I'm going to go ahead and erase this because you've got that handout and you know how it works now. And there is some basic anatomy we need to cover involving the eyeball. There is your eyeball. Now, and again, this will be your, we'll make this your left eyeball. So over here will be your nose. And over here will be your ear hole. And as you may or may not recall, I mentioned that there were going to be 6 muscles associated with the eye that you were going to need to learn. Remember, the eyes, let's try that again. The muscles that move the eyes through space are the only muscles in the body that are pure muscles, that are pure fast glycolytic, those fast twitch muscles. And the good news is, while we didn't learn them in the muscular system, they're going to be super easy to learn now. Because one is a straight muscle that goes towards the nose side of the eye. one is a straight muscle above the eye, one is a straight muscle below the eye, and one is a straight muscle on the ear hole side of the eye. And luckily, back in the last section, we learned about straight muscles. There was a straight muscle in the belly. There was a straight muscle in the leg. What do we call the straight muscles of the body?
Rectus.
Rectus. Now this rectus happens to be above the eye. Guess what we call this straight muscle above the eye?
Superior.
There you go. Superior rectus. Excellent. Guess what we call the straight muscle below the eye?
Inferior.
Excellent. The one towards the nose side of the eye?
Medial.
And the one towards the ear hole side? Excellent. See how simple that was? Now, I did say 6. So there happens to be a angled muscle under the eyeball that comes off the eye, but comes off at an angle. And one that comes off the top of the eye at an angle as well. And this one does something really interesting where it goes into this little connective tissue pulley structure. And then comes out the other side, changing the angle of which it pulls. What do you think we call an angled muscle above the eyeball?
Oblique.
There you go. And this one's above the eyeball, so it's the
Superior.
oops, superior oblique. And what does that make the one underneath? Oops, did it again. Excellent. See how easy that was? Took me longer to draw than it took you to learn them. Super easy. 6 muscles that move the eye in space. All of this activity of moving the eye through space is pretty darn important. So not surprisingly, we have not 1, not 2, but 3 cranial nerves that control all of this muscle. And I do mean all muscle of the eye. And those 3 nerves are 3. Oops, let's do it this way. 3, 4, and 6. Now, I know we've been going in order, but let's cheat and go with 6. Cranial Nerve 6. What is Cranial Nerve 6 called?
Is it the abducens?
There you go. Abducens. What does abducens sound an awful lot like?
Abduction.
To
Abduction.
abduct. What does it mean to abduct again?
Away from.
Move away from the midline.
Move away from the midline. Excellent. So which of these 6 muscles would abduct the eye?
Lateral rectus.
And that's exactly what the Cranial Nerve 6 does. It controls the lateral rectus. Now, staring me in the eyes, can you abduct both eyes at the same time?
Some people can.
Yeah, not many though, right? Not at the same time. Obviously, if I do that, I may be ducting my eye here. If I do that, I may be ducting my eye there, but I pretty much have to adduct the other one to do that, right? I can't really make both eyes go out at the same time. Can you bring both eyes inward?
Yes.
Yes.
Yeah, it's a lot easier to bring both eyes inward. It's a lot harder to get them both to go out. So obviously, this is not something that gets a tremendous amount of control and effect. Not surprisingly, this is the smallest of our cranial nerves, because we don't have a huge need to be able to turn our eyeballs out to the side. So this is our smallest cranial nerve, and it does one thing and one thing only. It controls our lateral rectus. Let's jump to 4. What is cranial nerve 4?
The trochlear nerve.
Trochlear nerve. Excellent. Trochlea. That sounds familiar to me as well. Where did we use trochlea before?
In the ear.
Not in the ear, although that's a good guess. Where else did we use trochlea? You're thinking cochlea, I think, in the ear.
Trochanter something is the bones.
We did have some trochanters, or if you remember, in my humerus, I have a trochlea, and then my ulnar has a trochlear notch that sits around it. And remember, the trochlea was that bow tie looking bow feature. And if you remember, trochlea means pulley. Well, if you notice, our superior oblique changes its angle by going through a little piece of connective tissue that holds it in place that we actually call the trochlea. So this little bit of connective tissue that holds the muscle up to the posterior part, to the superior and medial part of the orbit of the eye is actually called the trochlea. So guess what muscle controls, guess what nerve controls the muscle that goes through the trochlea?
The superior oblique.
Well, true. The superior oblique is controlled by the trochlea. Trochlear nerve controls the superior oblique. Excellent. That is a perfectly acceptable function for it. However, we could also say what it does. Because it's an angled muscle, basically what this does is it turns the eyes down and in. You may not have ever thought of the trochlear nerve or the superior oblique muscle of your eye ever before in your life. But back in ancient times, there were things called books. And before we were adapted to them by their smartphones, when you stared at a book for a very long period of time, as you were turning your eyes down and in, after about the 3rd hour of reading, you would start getting this tremendous pain right up here, kind of in the center in the bridge of your brow, right? You get this horrible, horrible ache and pain, this soreness in the area, right? Anybody ever experienced that before from staring at a tablet or a book or a textbook or a computer screen for too long? Yeah,
Yes,
absolutely.
right
Most
now.
people have. Why? Because this is fast glycolytic muscle that fatigues rapidly. And if you continue to use this to stare down and in at your computer screen or at your tablet or at your phone or at a book for a very long period of time, that muscle becomes fatigued and sore. It's one of the reasons why they always tell you to look up and look away at the distance every once in a while to let those muscles rest as a result of that. Excellent. So again, we can think about what it controls and what it does. Oh, I guess we could do that. We didn't do the same thing for 6. Let's go back to 6 real fast. If we think about what it does, we already mentioned it. AB ducts the eye. Oops, not AB sucks, AB ducts. Oh, and it should go without saying, but I'll ask it anyway. What do you think the functional type of Cranial Nerve 6 is?
Motor.
Motor. And of course, what is the functional type of the trochlear nerve?
Also motor.
Yeah. Perfect. So there you go. Cranial nerve 4, trochlear nerve, cranial nerve 6, abducens, and we have identified what they do. So what that leaves us with is cranial nerve 3. And what is the name of cranial nerve 3?
Ocular Motor.
There you go. Now, with a name like ocular motor, what do you think its functional type is?
Motor.
Motor, heck, it's in the name, absolutely. Now, again, we can think of it the same way. It controls, its job is to control basically all the other muscles. So it controls the superior rectus. It controls the inferior rectus. It controls the medial rectus. It controls the inferior oblique. And as some of you got sort of right on the last exam, some of you had the question, where might you find smooth muscle in the head? Well, inside the eyeball here, we have smooth muscle. And that smooth muscle does two things. One of the things it does is it affects the iris, changing how much light comes in. The other is to change your focus. So it changes the shape of the lens. To change your focus, so you can focus up close and focus far away. So this smooth muscle inside the eyeball is also controlled by ocular motor. So it also controls the smooth muscle inside the eye. So you can kind of see where it gets its name, ocular motor, because it controls most of the muscle, both skeletal and smooth muscle associated with the eyeball. Now, this is a very simple illustration, but your book, thankfully, has some very pretty ones. Notice here, we see these 3 nerves. Notice if we look closely, I'll do this. Notice here is 6 abducens, and notice it's coming up, and they've cut it, but you can see where it's controlling the lateral rectus so that it would abduct the eye. Here's cranial nerve 4 coming around and communicating with the superior rectus. Notice there's that connective tissue we talked about that changes the angle of the superior oblique. And then cranial nerve 3, ocular motor, goes to, notice it goes to the superior oblique, it goes to the superior rectus, inferior oblique, it goes to the medial rectus, and thanks to this ganglion, it goes to the smooth muscle inside the eye as well. So we've got their names, we've got their functional types, we've got their specific functions. Questions on any of that? All right.
So these ones don't exit the skull because they just stay there, right?
Well, but they do exit the cranial cavity to get to the orbit of the eye, just like the optic nerve leaves, but you absolutely have the right idea, right? Let's cheat and go back here, right? We know that this is the VIP entrance for the optic nerve only. So things like cranial nerve 3, things like cranial nerve 4, things like cranial nerve 6, they need more of a, general public type of entrance to be able to get in. So is there such an opening, a nice big large opening that would be easy for all 3 of those nerves, plus maybe some blood vessels and things like that, to get into the orbit of the eye and be the skull exit?
Yeah.
What would that be?
Occipital fissure.
Yeah, superior orbital fissure, absolutely. Which remember is a bone feature of the sphenoid bone. Absolutely. And in fact, if we look at the pictures, your book does a nice job of showing that. There's your superior orbital fissure. That's 4 coming through, and 6 coming through, and 3 coming through. But the last thing I want to point out is now we know their functions. They all have the same skull exit. Makes that super, super easy. So the real key is telling them apart. So let's start easy. 6. 6, as I mentioned before, is down here below the pons. As I mentioned, it is a very sad, very lonely, very pathetic little cranial nerve. It's the smallest, and it's kind of displaced away from its buddies that it goes to school with. Notice it's down here below the pons with 7 and 8. But are 6, 7, and 8 equally distributed away from each other? All equidistance?
No.
No.
No. 7 and 8 are BFFs. And poor little 6 is all here by his lonesome. So 6 is down here medially below the pons by his lonesome, away from 7 and 8. Notice 3 and 4 are above the pons. 3 is medial, and 4 is lateral. Although 4 is special. 4 looks lateral until you turn it around. When you look at the back side of the brainstem, what you see is that 4 actually comes off the back side of the brainstem. 4 is the only posterior cranial nerve. All the other cranial nerves come off the front, the anterior part of the brainstem. This is the only one that comes off the posterior. Now notice when we look at it from the front, it looks lateral, but it's really sneaking its way around from the back. So 6 is the smallest. 4 is the only posterior one. So these are kind of special that way, because it actually comes off the back side of it. So 3 is medial above the pons, 4 is lateral or posterior above the pons, and 6 is below the pons and medial. And again, I don't care how you describe them. You just on the exam, if I point to one of these, you need to know which is which is which. But knowing what's special about them is probably important too. Knowing which one is the smallest, which one is posterior, all right, which one comes off the telencephalon, right? Anything that's unique about these is probably a good thing to know as well. All right. Questions on that? All right. The good news is, remember, we have 12 cranial nerves, and we've already done 5 of them. So that means we only need 1 more to get halfway through. That's the good news. The bad news is the one that's remaining is the trigeminal nerve. And the trigeminal nerve's got a whole lot going on. And seeing as it's 4:25 already, I think this will be a good stopping point. We got through 5 of them. So what we will do Thursday after, let me write myself a note on that. After your 10-point brain anatomy daily quiz, after that 10-point daily quiz at the beginning of class, 10 questions, 10 points, we will then finish with our cranial nerves and go on from there. All right. Questions on any of that?
No.
Nope.
All right, excellent. Then you guys have a wonderful day. You know how the game is played. Start looking forward at the next ones. We will finish off. So we're right on schedule. Well, we're kind of not on schedule. Right on schedule for where we normally are, where we're behind. We will finish our cranial nerves. Then we need to finish our discussion of the spinal cord. And then we will start the autonomic nervous system on Thursday. So we should be able to do all those things on Thursday. Finish the autonomic nervous system maybe on Tuesday, and then that should give us a day for sensory stuff. And if we run behind, then we'll do less sensory and more autonomic. So we'll make it work. We're going to be there. We're going to get through all the important stuff. Whether we get to all the fun stuff or not, we'll see. But we'll definitely get through all the important stuff. All right. Any questions?
I have a quick question.
Yes.
When in the fall with 431, when we pick up, are you gonna start at the next chapter of the book?
I don't know what the next chapter of the book is. Here's what I will say is that you've got the right idea. We're going to cover the stuff that we haven't covered. In 430, the order is pretty much set. You obviously have to talk cells before you're going to talk tissues. You have to talk tissues before you can talk organs. It makes sense to do the bones before the muscles and so on and so forth. In 431, instructors vary more on what type of materials they cover. If you take me, we will do the cardiovascular system first, starting with the heart.
Okay.
So,
do we skip the endocrine system, or
we will do the endocrine system, but I don't do it first.
okay?
So, yeah.
Okay, I hear you. Thank you.
So yeah, everything we don't cover: endocrine system, cardiovascular system, lymphatic system, digestive, respiratory, urinary, reproductive. So the 7 systems we haven't covered, we will cover in 431. But everybody kind of does them in a different order. It doesn't have the same sequentialness to it that 430 does. So I've had a couple students already ask me about this. I will tell you the same thing that I told all of them. It is much, much easier to get into 431 than it is to get into 430. The problem that you're running into with 431 is that there's no limitations on someone signing up. So I guarantee you right now that at least 2 or 3 people in that 431 class right now are people who are signed up for 430 in the summer, thinking that they can complete all of this information you're completing in 8 weeks and be successful, and then take 431 in the fall. And I guarantee you most of them won't be successful. So there are plenty of people taking up spots. There are people who are on the border of 430 right now, hoping that they will get the grade that they need in this class. And I don't just mean mine, I mean in any 430 class. And so again, they're taking up spaces in the 430 classes, and they might not. There are students right now in 431 who aren't sure about their grades, so they're hedging their bets. So they're thinking, all right, if I have to drop this, I want to be able to take it in the fall. So they're signing up for the fall, and they'll pass. There is going to be tremendous turnover on the roster. So even if the roster is filled now, keep checking it, especially when spring grades come out. So the 3 big turnovers, 1 or 2 people will drop every day. But the 3 big turnover points are when spring grades come out, after the first week of summer school, because either people didn't get into the summer school class they wanted or dropped the summer school class they wanted, and then when the summer grades come out. So there's always going to be turnover, but those 3 times are huge times of turnover. There's going to be lots of turnover, and I'm certain that spots will open up. So if you're diligent, you keep checking the wait list, a spot will open up. And once you get on the wait list, you get in the first week, and usually, again, not always, but usually I'm able to get most of the students in that want to get in. So usually it's much, much easier to get into 431 than it is to get into 430. So keep checking, be diligent, and hopefully you'll get in. All right. The one thing I will warn you, and I was talking about students earlier as well, in the fall, there are going to be some 430 and 431 classes that will have labs on campus. Unfortunately, I was not picked to be one of those to be able to do that. So I will be fully online. So if you take me in the fall, we will be fully online. Well, there will be some instructors who will have labs on campus. Now, whether you're online or on campus, Open Lab with Jeff Chingaris will be available on Fridays. So even if you're fully online, on Fridays, you could go into the lab if you wanted to see the materials and the resources there. But my classes will be just like this. They will be synchronous. They will be online fully. So I am stuck here for the fall. So better or for worse. All right. Excellent. Any questions on any of that? All right. I'll see you guys Thursday.